In this video, we will discuss about one of the strategies that is used against the patient that have hemophilia. That strategy is popularly known as ITI strategy or immune tolerance induction. So what happens in this strategy is that uh, body has been temporarily uh, made into a condition tolerate where it can tolerate the proteins that lacks in hemophilia patient, that is factor 8 in hemophilia A and factor 9 in hemophilia V. So what happens in this ITI strategy is that uh, they will try to reduce the immune response against those proteins, and three kind of ITI therapies are been shown in this figure. First we will discuss with uh, the panel A, that is introduction of rapamycin. Introduction of rapamycin, it will activate the antigen presenting cells, and simultaneously what rapamycin will does is it will reduce the mTor signaling pathway. So so with the activation of antigen presenting cells, it will try to activate CD4 plus cells, and in order to completely activate CD4 plus cells and to convert them into effector cells, as we have discussed in our previous videos about immune response, that it needs two kind of signals. One is about the antigen, and uh, to another is to provide the present situation within the patient about the foreign substance. So that thing is been provided by mTOR signaling pathway. So however, in case of rapamycin, that decreases the mTOR pathway. CD4 plus will only receives one kind of signaling, that is about the antigen of rapamycin. However, it will not get enough signals of mTOR to convert CD4 plus cells into CD4 plus effector cells, and this leads to the apoptosis of CD4 plus cells. And simultaneously, this rapamycin will induce Treg pathway, and this Treg pathway is responsible for reducing the immune response, such as it will prevent the activation of CD4 plus cells into effector cells, and it will reduce the adaptive immune response also. So how this therapy works in this case is that introduction of rapamycin and factor 8 simultaneously for a short duration of introduction of rapamycin and factor 8 simultaneously within the patient at shorter dosage will help the will help the factor 8 to remain in the patient for a longer duration of time. So the second strategy is using these leaves. These leaves are designed in a way that it will also reduce the CD4 plus will also reduce the CD4 plus uh, activation into the effector cells, and simultaneously it will uh, induce this Treg pathway. So there are two kind of Treg pathway that are introduced in case of this uh, consumption of these leaves. So that Treg pathway will leads to decrease the response of adaptive immune response and the conversion of CD4 plus to CD4 plus effector cells. So, and simultaneously this FOXP3 Treg pathway will also get activated. It will also prevent the it will also this Treg pathway will prevent the maturation of CD4 plus T cells, and direct these CD4 plus T cells towards cell death or programmed cell death or apoptosis. The third pathway is the latest one, that is using associated virus with or lentivirus with promoter or miRNA, or, or we can say factor 8 gene of interest within this adeno-associated virus. And what this will does is uh, this will also leads to a production of more factor 8 virus, and simultaneously it will also contain the miRNA target, or target to target to decrease the activation of CD4 plus T cells, and simultaneously promote or induce this FOXP3 Treg. So the both of this will reduce the maturation of CD4 plus T cell into the effector T cell, and simultaneously this induction of this FOXP3 will FOXP3 will reduce the immune response, or we can say adaptive immune response against the patient. And all these things are introduced or are taken at a uh, lower dosage uh, after a short duration of time, so that the proteins that are proteins that lacks in this hemophilia patient can stay or have a high half-life period within the patient. So that's all for this video. If you need to understand more about this immune tolerance induction, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.